Good afternoon, everyone. I'm Bill Kennedy, chairman of the Global Interdependence Center, and welcome to our new series, Long COVID, Research, Policy, and Economic Impact. Today's program is being recorded and a replay will be posted to the GIC website starting tomorrow. Throughout 2022, GIC, in partnership with the Solve Long COVID initiative, will host webinars and in-person conferences, bringing together world-class experts to explore long COVID's impact on science, medical research, public health policy, labor markets, and the implications for fiscal and monetary policy. This special series is made possible in part by the David R. Kotok Global Healthcare Series, an endowed program established in 2020 by a generous lead donation from longtime GIC program chair, director, and friend of the GIC, David Kotok. David, thank you. So before introducing the panel, I would like to invite Ovid Amate, Chief Executive Officer of Solve ME, to say a few opening remarks. Ovid?
Thank you very much, Bill. I'm happy to be here today. It's been only a couple of months ago, after nearly a year and a half of a global pandemic, that the World Health Organization published for the first time a definition of long COVID, calling attention to one of the most concerning yet under-recognized aspects of the COVID-19 pandemic. In fact, it's not unusual for people to develop long-term debilitating symptoms after infection by a wide variety of viruses and bacteria. As Dr. Anthony Fauci said recently about what people experience after contracting COVID-19, he said, it's extraordinary how many people have a post-viral syndrome that's very strikingly similar to the illness known as myalgic encephalomyelitis, that's often referred to as chronic fatigue syndrome or ME/CFS. And he said, they just don't get back to normal energy or normal feeling of good health. So based on more than 30 years of research and advocacy experience in this space, Solve ME was one of the first organizations to recognize the emerging threat of the long-term debilitating outcomes of the global COVID-19 pandemic. As early as April of 2020, we were preparing to embrace the COVID long haulers, and we called on Congress to have an immediate response measures. Since then, we felt compelled to expand our programs based on the premise that the significant body of knowledge and experience of the ME/CFS community will benefit people with long COVID, and at the same time that research into long COVID could have a positive impact on people who previously had an existing disease. The COVID-19 pandemic is a tragic in so many ways, but it is also an opportunity that we must not waste. So this collaboration with the GIC takes our work to the next level with the hope that 2022, then in 2022, we can make progress in addressing the needs of people with post-infection diseases worldwide. So again, Bill, thank you for the opportunity to work together, and let me turn it back over to you to get us started with today's program.
Terrific, thank you, Ovid. So why are we doing this? Well, there are nearly 300 million reported COVID cases worldwide. How many of these people will be struck with long COVID? No one knows for sure, but consider if in the coming decade, 10% or even 5% of the labor force were chronically disabled. Imagine the implications for the medical system, health policy, the insurance industry, the labor markets, and even geopolitics. So our purpose for this series is to explore the many interconnected and interdependent threads of long COVID. And that brings us to today's presentation. So it's a distinct honor to introduce Dr. Amy Proal, a microbiologist at Polybio Research Foundation, an organization committed to studying chronic inflammatory diseases. As a member of the research team at the foundation, her expertise is on molecular mechanisms by which pathogens modulate and impact human gene expression and metabolism and immunity. Dr. Proal has authored numerous research papers and presented at conferences around the globe, and she has lectured at the National Institute of Health. She earned her degree in biology from Georgetown University and has a PhD in microbiology from the Murdoch University in Australia, where she successfully defended her thesis, Autoimmune Disease Reexamined in Light of Metagenomic Concepts. Moderating today's discussion is Emily Taylor, who's the Vice President of Advocacy and Community Relations for Solve-ME. She brings over 15 years of policy and organization and advocacy experience in both the nonprofit and government sectors, including 5 years working on Capitol Hill. Emily earned her degree in politics and international relations from Scripps College in Claremont, California, where she graduated with honors, and she earned a master's in American politics from Claremont's Graduate University. Emily draws inspiration from her mother, who has battled chronic autoimmune conditions and ME and chronic fatigue syndrome for over 2 decades. So please join me in welcoming Emily and Dr. Proal. I'll turn the floor over to you.
Thank you so much, Bill, and thank you, Ovid. I'm looking forward to our discussion later on in the program and answering the questions from the audience. With that, we'll begin the presentation. Amy, what can you tell us about long COVID and its impact on the biomedical and scientific fields?
Hi, well, thank you so much for having me to speak today, and I'm going to share some slides to help address that question. So let me do that. Okay, so today I'm going to discuss biological factors that may contribute to the development of long COVID. Now, as I was already described, we're here today because across the globe, millions of patients are developing a range of debilitating chronic symptoms that do not go away. In fact, studies indicate after COVID-19 infection. 1 second, I'm just going to fix something with my screen. Okay. Hold on. Okay. In fact, studies indicate that up to 30% of patients who get COVID are developing persistent symptoms. And these patients are being given the diagnosis long COVID or post-acute sequelae of COVID or PACS, which I'm just going to call long COVID from now on. And the graph on my slide lists just some of the symptoms being experienced by long COVID patients. And importantly, long COVID is being diagnosed in patients who develop severe acute COVID, but also in patients who developed only mild or even asymptomatic cases. So, while the development of long COVID or chronic symptoms after COVID is sometimes framed as novel or mysterious, it is actually a well-recognized phenomenon. So over the past decades, most well-studied viral or bacterial pathogens have been connected to the development of chronic symptoms in a subset of infected patients. These include Ebola, Zika, dengue, and bacterial pathogen Borrelia burgdorferi. And we refer collectively to these diagnoses as infection-associated chronic disease. For example, before the pandemic started, we were already studying myalgic encephalomyelitis, or ME/CFS, a chronic condition in which patients develop debilitating chronic symptoms after other viral infections. For example, after infection with the herpes viruses or enteroviruses. And enteroviruses, like SARS-CoV-2, are single-stranded RNA viruses that include the Coxsackie viruses, the echoviruses, and the rhinoviruses that can also be acquired via respiratory infection. And interestingly, there is great overlap between the core symptoms of ME/CFS and long COVID. In both conditions, symptoms include flu-like symptoms, symptoms that can relapse and remit, post-exertional malaise, or great difficulty recovering from even minor bouts of exercise. Also, autonomic dysfunction. Patients have blood pressure issues, heart rate issues, and great difficulty moving from a sitting, standing to standing position without getting very dizzy. So when long COVID cases started to be reported early in the pandemic, we quickly pulled from our research on ME/CFS and related infection-associated chronic conditions to write this review on biological factors that may contribute to persistent symptoms in patients with long COVID. And in the paper, we describe at least 7 central factors that may contribute to long COVID development, none of which are mutually exclusive, or they're all connected, and different patients may have one or more issues occurring at the same time. And these are organ injury. A person may have experienced severe inflammatory disease during acute COVID and they still may have not recovered from that, especially in the lungs. Next is the straightforward possibility that at least some patients with long COVID may not have fully cleared the SARS-CoV-2 virus from every body site, and it remains in a low-level tissue reservoir where it continues to provoke the immune response. We call that viral reservoir. Another possibility is that immune dysregulation during acute COVID allows other pathogens already harbored by the patient, such as herpes viruses, to reactivate and themselves drive chronic symptoms. Also, a similar trend can happen with the body's microbial and viral ecosystems, the human microbiome environment. During immune dysregulation during acute COVID, they may collectively shift towards a state of imbalance that can also drive chronic symptoms. Finally, platelet hyperactivation, blood coagulation, and extended autoantibody production, often via molecular mimicry, which I'll explain more about later, may contribute to symptoms in patients with long COVID. And last, we think the disruption of vagus nerve to brainstem signaling may account for some of the core symptoms experienced by patients with the condition. So the topic of organ injury in long COVID is fairly straightforward. So I'm going to start by discussing the possibility that at least in some patients with long COVID, the virus may not fully have cleared from all body sites. And there is growing evidence that SARS-CoV-2 is capable of persistence in a range of human tissues and cell types. This is a preprint from a large recent NIH autopsy study. And the team identified SARS-CoV-2 in a very wide range of human body sites, from skeletal muscle to adipose tissue to the cervical spinal cord and regions of the brain, including the brainstem, basal ganglia, and cerebellum. And some of the subjects in the study harbored the virus in a persistent state for an extended time, well over 30 days. In one case, for up to 230 days, which was the end of the study. And it's worth noting that most of the patients in this study were hospitalized and they had also been given systemic steroids. But if the findings hold in peer review, the study serves as an excellent proof of concept that SARS-CoV-2 is capable of persistence in not just the body, but also the brain. So other teams have reported similar findings in tissue samples obtained from living patients. For example, this study in the British Medical Journal identified SARS-CoV-2 nucleocapsid protein in viral RNA in colon, appendix, ileum, hemorrhoid, liver, gallbladder, and lymph node tissue collected during surgery from 5 patients 9 to 180 days after acute COVID disease. And here's another example. This team found either SARS-CoV-2 protein or RNA in intestinal tissue samples collected via colonoscopy from patients about 4 months after acute COVID. And the patients were all asymptomatic at the time of tissue collection. And because intestinal tissue is a very likely reservoir for SARS-CoV-2, we're in the process of helping this team try to expand this research to a larger cohort of long COVID patients. And by the way, chronic enterovirus infection, those single-stranded RNA viruses I mentioned, have been documented in intestinal tissue from patients with ME/CFS. This team obtained intestinal tissue via endoscopy from 165 patients with ME/CFS. And they found that 82% of specimens stained positive for enterovirus VP1 protein compared to just 20% of controls. And viral RNA was also found in some of the tissue samples. And so I want to pause here and talk about a key consideration. In all the studies I just described on SARS-CoV-2 persistence, patients tested negative for SARS-CoV-2 via routine nasal swab testing. In fact, at the time that their tissues were collected for study. And that feeds into a general rule about pathogen reservoir in infection-associated chronic disease. Generally, pathogens tend to clear from body fluids such as the blood, urine, and secretions over time. And instead, in simple terms, they hide in patient tissue or nerve, where they're less likely to be targeted by the immune system. And it follows that to correctly study the topic of SARS-CoV-2 persistence in long COVID, we must study tissue samples from patients with the disease. The study of blood alone is not going to get us there. And to make that happen, we are collaborating with clinicians to collect tissue samples from long COVID patients that can be obtained during routine biopsy or surgical procedures. And we're working with an incredible research team from the J. Craig Venner Institute to create a pipeline that will use whole genome sequencing, spatial transcriptomics, and other methods to analyze those tissue samples, not just for SARS-CoV-2, but for a range of other organisms and immune cell abnormalities. Now, one thing though, is that even if SARS-CoV-2 does not itself persist in the blood, the proteins it creates can leak into the blood, where they can drive serious pathology. For example, Alessio Fasano and team at Mass General studied children with multi-system inflammatory syndrome, or MIS-C, which is a serious inflammatory condition with parallels to long COVID that can occur in children. And they found that in children with MIS-C, SARS-CoV-2 did not clear, but remained in the gut for weeks after initial infection. And this led to the release of zonulin, which is a biomarker of intestinal permeability, with subsequent trafficking of the SARS-CoV-2 spike protein into the blood, leading to hyperinflammation. And it's possible that a similar phenomenon could occur in at least some patients with long COVID, which is bolstered by the fact that the SARS-CoV-2 spike protein can catalyze clotting or coagulation in blood. And in this very important long COVID study, Dr. Ricia Pretorius and team at Stellenbosch University in South Africa used techniques such as fluorescence microscopy to demonstrate the presence of circulating persistent amyloid fibrin microclots that are resistant to fibrinolysis or normal breakdown in blood collected from long COVID subjects and not in controls. In separate experiments, they have shown that the SARS-CoV-2 spike protein can catalyze formation of these microclots, which is accompanied by platelet hyperactivation, platelets being tiny blood cells that contribute to clotting processes, which can then lead to the release of inflammatory cytokines and other inflammatory molecules that damage the vasculature or the body's blood vessels, which I'll come back to later. Another factor that may contribute to persistent symptoms in some long COVID patients is that SARS-CoV-2 may fully clear from a patient after acute infection. However, the virus may dysregulate the host immune response during acute COVID in a manner that allows previously harbored pathogens to reactivate, infect new body sites, and themselves drive new chronic symptoms. And this is possible because most humans accumulate persistent viruses across the course of a lifetime. In fact, the vast majority of people harbor multiple strains of herpes viruses. And a more detailed example of that are the findings displayed on this slide from a Japanese study. The researchers took RNA-seq data from the Tissue Expression Project, a public resource created to study tissue-specific gene expression and regulation from 51 tissue types collected from hundreds of healthy individuals at autopsy. So these were people that died, unfortunately, from accidents, but not from illness or related events like accidents. And they successfully identified 39 viral species in a range of tissue types, including tissue from the blood vessel wall, the pituitary gland, and the ovaries. And identified viruses included DNA viruses like the herpes viruses, but also a range of RNA viruses, including, interestingly, some coronaviruses, not SARS-CoV-2, but previous coronaviruses. So what does that mean for long COVID? Well, under conditions of health, these viruses are kept in check by the human immune system. The immune system has defenses that keep them in a largely dormant or inactive state. And this is a well-known phenomenon. Under conditions of stress or immune dysregulation, the same viruses can reactivate and begin to drive symptoms. And now consider that infection with SARS-CoV-2 dysregulates the immune response. For example, the virus expresses at least 10 proteins that disable host interferon signaling. And interferons are proteins that control viral activity and replication on a number of levels. So when their activity is knocked down, it is much easier for viruses like the herpes viruses to emerge from latency. And SARS-CoV-2 infection during acute disease can also deplete T cell activity, which can even further facilitate herpes virus reactivation. And also, I just want to clarify, it doesn't just have to be viruses that may reactivate in some patients with long COVID. Some patients may harbor bacterial pathogens, such as the tick-borne pathogens, Borrelia or Bartonella, or even parasites like Toxoplasma that can drive neuropsychiatric symptoms when activated. So studies are beginning to demonstrate that pathogen reactivation is indeed happening in some long COVID patients. For example, in this study of 30 long COVID patients, they found that about 67% were positive for Epstein-Barr virus reactivation, compared to about just 10% of the control group. And these were subjects who had gotten COVID. The control group were patients who had gotten COVID but didn't develop persistent symptoms. And the images on the slide show skin manifestations of this Epstein-Barr virus reactivation in the long COVID subjects, although EBV, Epstein-Barr virus, can reactivate without skin symptoms. And interestingly, 4 of the long COVID patients in this study were asymptomatic at the initial time of acute COVID disease. So when this activation occurs, what happens? Well, for one thing, the reactivated pathogens may begin to express proteins that their latent counterparts largely do not. And these proteins can directly bind into host receptors to modulate human gene expression, the host immune response, and host metabolism. Or the pathogen can simply infect a new body site, tissue, or nerve in a manner that allows it to drive new chronic symptoms. And if it's able to get into the central nervous system, it can really start to cause problems. For example, this team found that Epstein-Barr protein, EBNA2, directly binds half of the locations on the human genome that contribute to the lupus risk. So the virus is amplifying human gene expression. It's modulating risk of the disease. And we need to think about that trend more in long COVID. In addition, I wrote this paper this past year, along with my PolyBio Research Foundation colleague and Harvard neuroscientist, Mike Van Elseker. And our goal was to connect key trends in the immunometabolism research community. And we describe how viruses and many bacteria are obligate intracellular pathogens. And what that means is that they rely entirely on the metabolic capacity of the host cells they infect to gain raw materials for their replication and nutritional needs. So to obtain them, they hijack the mitochondria of the cells they infect to redirect glycolysis and TCA cycle intermediates towards the synthesis of lipids, fatty acids, and nucleotides for their own replication purposes. And that by definition changes the metabolic output of the infected cells. And interestingly, mitochondria are now being studied as part of the innate immune response that combats that hijacking. And overall, this matters because teams have reported metabolic abnormalities in patients with ME/CFS, and now to a point in patients with long COVID. But we can begin to connect those abnormalities with the pathogens and organisms also tied to the disease process. And okay, what else can reactivated viruses do? Well, remember the study, the long COVID microclots identified by Ricia Pretorius and team? Well, remember that platelet hyperactivation contributes to microclot formation. This is a diagram of a platelet taken from another of Ricia's papers, and it shows that platelets have receptors that recognize a range of viruses or their proteins. In fact, they activate in response to herpes viruses, enteroviruses, or other viruses that may reactivate in patients with long COVID. And that activation is part of a normal immune response. But if the infection or associated inflammation does not fully clear, the platelets can become inappropriately hyperactivated in a manner that contributes to clotting, release of inflammatory cytokines, and possible vascular damage, with the takeaway being that reactivation of herpes viruses or related pathogens in patients with long COVID can also contribute to or augment the clotting that Ricia and team are identifying in long COVID blood. And because many of these same viruses are implicated in ME/CFS, we are working with Ricia and team to extend this study to a cohort of patients with ME/CFS. Now, it's also very important to understand that pathogen activity can contribute to autoantibody production in patients with long COVID. And what I mean by that is autoantibodies are often generated due to molecular mimicry between pathogen and host proteins. And to explain that further, when human antibodies are notoriously polyspecific, they have flexible specificity. In all major pathogens, SARS-CoV-2, the herpes viruses, they express proteins, and those proteins often are very similar in size and shape to human receptors, human proteins, or human tissue. So when the immune system fires on the pathogen protein, it may target human tissue with a similar structure. It cross-reacts. And what we call the antibody that does that is an autoantibody. So here's just one example of that, proof of concept. This team found functional G protein coupled receptor autoantibodies in long COVID subjects. In cytomegalovirus, which is one of the herpes viruses that has been shown to reactivate in some long COVID patients, creates proteins that are known homologs of the G protein coupled receptors and chemokines. In other words, that's similar size and shape. So overall, we can increasingly begin to ask, can there be an infectious origin to the generation of the G protein coupled or other autoantibodies identified in patients with long COVID? Now, with that in mind, it's also worth considering that imbalances of the body's microbial ecosystems, the human microbiome environment, may contribute to long COVID. Humans are not sterile, not by a long shot. We are filled with trillions of interacting communities of organisms, bacteria, viruses, fungi, archaea. And now we know that not just the gut and the mouth, but most of human tissue and blood are filled with these interacting organisms. There are even viruses called bacteriophage in these communities, and they infect bacteria and modulate their activity. And there are 10 times more bacteriophage in any microbiome ecosystem than there are bacteria. So Paul Bolsky, who's a researcher at Stanford, estimates that there are about a quadrillion bacteriophage trafficking through human tissue and blood at any given moment. And here's an example of how expansive bacteriophage populations can be in the human body. This past year, I worked with a team at the Berkeley Joint Genome Institute, and we assembled the metagenomic gut virus catalog that comprises publicly available human stool metagenomes. And based on sequence clustering data, we identified 54,118 candidate viral species, 92% of which were not found in existing databases. In other words, they were not known to exist before we performed this study. And with that in mind, I cannot impress on you how impossible it is to fully study any chronic condition without taking the activity of these organisms into consideration. And how does that fit then into the pathogenesis of long COVID? Well, it's a little like the latent viruses. Under conditions of health, host microbiome and virome communities are kept in check by a robust human and host immune response. They persist in a state then of balance or homeostasis. But most organisms of human microbiome communities are what are called pathobionts. They can act as commensals under conditions of health, or they can change their gene expression to act as pathogens under conditions of inflammation and imbalance. For example, E. coli. E. coli is a normal member of many microbiome ecosystems, including the gut. But one team showed that in less than 500 generations, E. coli can change its gene expression to act as a virulent pathogen that can drive severe disease. So, under conditions of acute COVID disease, where SARS-CoV-2 dysregulates the immune response, the body's microbial and viral ecosystems may collectively shift towards a state of imbalance and pro-inflammatory gene and metabolite expression that can drive a broad range of chronic symptoms. And here's an example of that being documented in long COVID. This team found that in long COVID patients, about 10 weeks after acute COVID, they had significantly higher abundances of oral microbiome species that are known to promote inflammation via LPS production, with LPS being a component of the bacterial cell wall, or other mechanisms. And they found a reduction in anti-inflammatory metabolic pathways. And the flow-on effects of that dysbiosis can lead to dysfunction of oral barriers, such as the gingiva, which allows some of these organisms and the LPS to leak into the blood. And this same leakage may occur from other body sites, the gut or other places. And remember this study, the microclots that Ricia Pretorius and team found in long COVID blood? Well, the platelets that hyperactivate as part of the microclotting process also have receptors for a wide range of bacteria and bacterial products like LPS. So microbiome and virome dysbiosis in any long COVID body site and subsequent leakage of the bacteria or LPS into the blood can also augment coagulatory and vascular issues in long COVID patients. And one consideration is that if the blood becomes more coagulated, more clotted in long COVID, either due to the virus itself or contributing impact from reactivated viruses or microbiome dysbiosis, or a mix of the factors, then the clotted blood will have more trouble perfusing the body, especially the smaller capillaries. And that could contribute to the development of small fiber neuropathy, which is a condition in which the body's small peripheral C fiber nerves become damaged or diminished. You can see an image of those damaged nerves on my slide. And that is because the blood perfusion via the capillaries is low. The energetically demanding C fiber nerves may not get the blood supply that they need, and they may then become damaged. And that is being documented. It is documented often in ME/CFS, and it's being documented now in long COVID, for example, in this case series by our colleague, Peter Novak. So because the blood carries oxygen with it as it perfuses tissue, impaired blood flow due to clotting may lead to hypoxia, or low oxygen levels in tissue of patients with long COVID. And this team did find that in long COVID patients, they exhibited impaired systemic oxygen extraction, along with marked reduced peak exercise aerobic capacity. So there were signs of possible hypoxia. And hypoxia in turn may aggravate other aspects of the disease. For example, hypoxia has been shown to induce Epstein-Barr virus reactivation via the mechanisms described on this study on my slide. Now, additionally, if blood flow or the vasculature becomes compromised in patients with long COVID, that could lead to problems with blood perfusion in the brain in a manner that might contribute to cognitive symptoms or brain fog. And that's not a stretch, because my colleague Mike Van Alzaker may be picking up on some problems with ME/CFS vasculature in an fMRI imaging study that he's running. And here are some of the data from that ongoing study. These images show a healthy control subject getting their brain scanned during a mild autonomic challenge. The challenge increases carbon dioxide in blood. It's a breath hold challenge, which should cause blood vessels in the brain to expand or vasodilate. And you notice that in the healthy person in this slide, the left and right images are the same because the brain's blood vessels appropriately regulate their response to this mild autonomic challenge. But these 4 examples show ME/CFS patients. You can see that the images on the left look fairly normal. They're taken during rest, but on the right, the blood vessels are failing to react like they should during the mild autonomic challenge. And importantly, not every patient is reacting in the exact same way. And now we are also working to extend this study to long COVID. And last, I want to discuss how disruption of vagus nerve to brainstem signaling may contribute to some of the core symptoms experienced by long COVID patients. And by core symptoms, I mean the flu-like symptoms, the sickness symptoms, and the autonomic dysfunction, the pain, and the nausea. To explain further, the vagus nerve is a very important cranial nerve that innervates every major trunk or organ of the body. And in all of these body sites, vagus nerve chemoreceptors can sense inflammation. And this inflammation can be generated in response to most of the factors I've described thus far, in response to ongoing infection, pathogen reservoir, or inflammation generated as a response to microbiome dysbiosis or reactivated viruses. And when the vagus nerve chemoreceptors sense this inflammation, they convey a pro-inflammatory signal to the dorsal brainstem, which is a core area in the back of the brain. And it just so happens that tightly clustered nuclei in a small overlapping region of the dorsal brainstem hold control over sickness, pain, nausea, and autonomic symptoms. So for example, this figure, which is included in our long COVID paper, shows these overlapping brainstem nuclei. And the feelings of being sick and nauseous are controlled by the nucleus of the solitary tract, or NTS, and the area postrema, AP. Inflammatory pain signaling is controlled by the dorsal reticular nucleus, or DRT. Autonomic functions are controlled by dorsal motor vagus, DMV, and nucleus ambiguous, NAMB. And these overlapping nuclei normally rely on precise signal-to-noise signaling. But when an inflammatory signal via the vagus nerve enters the brainstem, it activates glial cells there. And when glial cells, such as microglia, activate, they produce mediators that excite neurons, such as glutamate, ATP, and cytokines. And that means that glia act as an amplifier for neurons and can disrupt their precise signal-to-noise signaling. Therefore, when the dorsal brainstem is activated by pro-inflammatory signaling from the vagus nerve, the structures involved in sickness, pain, and autonomic function often become overly responsive, potentially leading to increased sickness, pain, nausea, and autonomic symptoms. And we are indeed running a PET study with Mike Van Elseker at the Harvard-MIT Martino Center, where he uses the radioligand PBR28 to measure microglia activation in subjects' brains, which is a proxy for neuroinflammation. And here is a heat map of PBR28 uptake in 2 subjects, one with ME/CFS and one with long COVID. And the brighter colors like yellow indicate more PBR28 uptake and thus more microglia activation. And in both subjects, microglia were more activated in the dorsal anterior cingulate cortex versus age, sex, and genotype matched healthy controls. And that's an important structure in the cognitive control circuit. Therefore, neuroinflammation in this area is a likely source of brain fog. And also, once activated, microglia can become primed. And what that means is they enter a new functional or behavioral state in which they respond more robustly to a subsequent challenge. And because microglia are long-lived, this prime state can last a long time. Indeed, this priming of glia may be part of why patients can become increasingly sick with subsequent infections or inflammatory events, what I sometimes call multiple hits. So how does this play out in actual patients with long COVID? Well, for example, let's say a person has SARS-CoV-2 reservoir in intestinal tissue. The vagus nerve can sense inflammation from that ongoing infection, convey a pro-inflammatory signal to the dorsal brainstem, and that could disrupt brainstem nuclei signaling in a manner that results in sickness, pain, nausea, and autonomic symptoms. In addition, protein created by the virus could leak into the blood, causing problems with coagulation, the vasculature, and hypoxia and tissue. And this person could be diagnosed with long COVID. Another patient could have fully cleared SARS-CoV-2 from all body sites, but Epstein-Barr virus could have reactivated, and they also suffer from severe microbiome dysbiosis. The vagus nerve can also sense inflammation from those 2 issues, convey a pro-inflammatory signal to the dorsal brainstem, and that could disrupt brainstem nuclei signaling in a manner that results in sickness, pain, nausea, and autonomic symptoms. In addition, protein created by the virus may leak into the blood, causing problems with coagulation, the vasculature, and hypoxia in tissue. And this patient could also be diagnosed with long COVID. Another person might have oral microbiome dysbiosis, SARS-CoV-2 reservoir in the lung, and enterovirus in the stomach. And these multiple hits will activate vagus to brainstem signaling even more profoundly, resulting in even more sickness, pain, nausea, and autonomic symptoms. And the takeaway is that these examples could go on. The takeaway is that if the same circuitry is being impacted, no 2 long COVID patients have to have the exact same mix of pathogens or inflammatory issues to develop these same sets of common symptoms. And with that, I have a couple more examples. Can I have 1 more minute?
Please,
okay.
Go ahead.
I just wanted to quickly explain that the most straightforward way that the vagus nerve to brainstem signaling could be disrupted in long COVID is if the vagus nerve is directly infected. Mike Van Elseker did write a paper in 2013 suggesting this may be happening in patients with ME/CFS with the herpes viruses. There is some preliminary evidence that SARS-CoV-2 can infect the vagus nerve. This German autopsy study did find SARS-CoV-2 in the brainstem. It was an acute COVID autopsy study, and also in cranial nerves, the vagus nerve. And an ME/CFS autopsy study found enterovirus RNA directly in the brainstem. So for what it's worth, in theory at least, another patient could have SARS-CoV-2 in the brainstem and direct infection of the vagus nerve by something like human herpes virus 6. And that would be one of the most straightforward ways to disrupt vagus nerve to brainstem nuclei signaling in a manner that could result in sickness, pain, nausea, and autonomic symptoms. And this patient could also be diagnosed with long COVID. Thank you.
Wow, thank you so much for that insightful presentation and really informative. Just so much to unpack here. So let's dive right in. We'll continue to the Q&A portion of today's event. I'd like to remind our attendees that you can enter your questions for Amy into the questions tab on the dashboard. So the first question I have was asked about 30 different ways and from 30 different people in the pre-registration questions. And I'll highlight just 2 of them. So the overall question is, are there potential subgroups of long COVID? I'm so sorry for my dog. I'm so sorry. I so apologize for that. I think there's someone at the door. So, so the question is about the potential subgroups of long COVID, and they pointed to a couple different pieces of evidence. One, that women specifically have been found with some evidence that are more susceptible to long COVID, and others about that there are some studies that are coming out of Asia that seem to show higher rates of long COVID. So, so do we have any evidence or theories about why certain demographic groups or certain subtypes might be more susceptible to long COVID?
Yes, I mean, there's a lot of factors that would feed into that question. I think first, in terms of just subgrouping long COVID patients, I think one of the points that I was trying to make with my presentation was that there may be less discrete subtypes and more of a continuum in which different issues are happening in different patients, but all of them impact or do common things. For example, like I was trying to explain it, the vagus nerve to brainstem circuitry is impacted. It could be a different virus in a different person, but patients might develop common symptoms, right? So not everyone has to have the same thing going on to develop long COVID symptoms. That being said, you can still, there's different factors that may predispose to long COVID. One thing that I didn't get a chance to discuss is that obviously our human genomes also have an impact on all chronic disease risk, right? So there may be certain human genome variants, which may differ among patients, that may also help determine how much any of the environmental issues that I focused on in my talk may be able to occur. For example, just as a basic explanation, there are some genes that predispose people, some variants in genes, to increased clotting issues. So if you already had human genetics that were more predisposed to you having clotting issues in blood, and then you also got sick and the SARS-CoV-2 spike protein catalyzed microclots in your blood, you might have a particular problem with that clotting, right? So in other words, there may be human genome variants that we can increasingly identify that will help understand why some long COVID patients have particular problems with certain parts of the disease process. And beyond that, the question of why more women than men have ME/CFS and long COVID. So that is, to me, a complicated question, and probably in some way comes down to the fact that the hormonal system, the endocrine system, and our immune systems are intricately connected. So that's some of what I did my graduate work on, is that the nuclear receptors that control endocrine or hormonal signaling in the human body, that control estrogen activity, androgen signaling, have immune components of the immune system under their control. So there's a direct relationship between your hormone signaling and your immune signaling. And of course, women's hormonal signaling is totally different than men, and it fluctuates more because of menstruation and pregnancy and events like that. So there may be, I think it's a promising line of research if we sort of look at the relationship, those core relationships between endocrine signaling and immune signaling, to better understand where there may be some patterns in that.
Fascinating. Thank you so much. That is a really detailed answer, and hopefully that answered a lot of different questions out there from a lot of folks. The next question I'd like to get into is about treatments. We've received a lot of questions about what is being done for treatments and the future, potentially in various drug markets or various druggable targets for treatments. So overall, why is the progress in treatments in this area been so slow? And what would you point to as medical factors for the slow progress of treatments?
In terms of treatment, I can't totally speak to why it's slow because I'm not an expert in the regulatory process with treatment. I suspect that there's, it's difficult to get drugs trialed. Trials are very expensive. There's a lot of coordination and a lot of work that has to go into those aspects of it. I do think, though, that we need to do the research to really understand. We're still in the stage in which we're doing the research to understand what's really happening with patients with long COVID. And when we have more clear data on that and more very solid and replicated publications on possible, the trends that I talked about, but better delineated, that's going to help with treatments, because we can obviously then pull from that to say, what can we do about it? So, if SARS-CoV-2 doesn't fully clear in a patient and there's viral reservoir, maybe we can use certain antivirals. If the clotting and the microclots, which are being identified in a growing number of patients with long COVID, if that continues to hold, then maybe we can use, or the team has already published using anticoagulants in acute COVID to prevent that microclot formation, or even using apheresis, which is a tool that removes clotting and other processes from blood on patients. So I think that what's key is that we really fund and accelerate research right now so that we can get to these core trends that we can quickly iterate into potential treatment options.
Thank you so much. I think that was a very popular question as well, and I'm sure there's going to be a lot of follow-ups. Another question that was shared is, so what is your opinion, Amy, about a COVID mutation evolution that triggers an acute cytokine storm in the victim of the infection and raises the COVID fatality rate? Is that something that could be on the horizon based on the studies that you've seen?
Yeah, I mean, that's a tough one. And I think is the question if a variant could arise that could be even more pathogenic and cause more of a cytokine syndrome. I think that's on the table. It's hard to predict how COVID is mutating. I mean, you can to an extent, but for example, the Omicron variant came a little out of left field. It's diverged from the original Alpha strain of COVID. It's not even diverged from Delta and its ancestors, and that's the dominant variant now. That was hard to predict. So there's a lot of variables that go into this. There's a lot of study of, how viruses mutate. There's certainly selection pressure when viruses mutate to become more infectious, to increasingly infect more people. We're seeing that with Omicron. Now, whether eventually there will be selection for pathogenicity, increased pathogenicity, I guess it's an open question. I hope not, but we can't say no.
Of course, absolutely. And I think it's especially given the rapidly evolving nature of the field, it's really hard to pin any of those items down. But thank you so much for sharing your thoughts and opinions on those potential outcomes. Another question that's come from the audience is about postviral myocarditis. I hope I'm pronouncing that correctly. Mm-hmm. The questionnaire points out that 1 in 5 adults with long COVID have this postviral myocarditis that can be found up to 3 months post the infection. Is this something that we need to raise awareness about? And is this something that that the research has confirmed?
I'm not sure of the exact numbers, but certainly I've heard of myocarditis developing in patients with COVID. Myocarditis being infection of the heart that can lead to severe cardiac symptoms. I think it's worth considering the reactivated viruses, that trend that I mentioned in which the immune dysregulation during COVID can cause herpes viruses or other already existingly acquired viruses to reactivate. It's known that many of the herpes viruses can drive myocarditis. It's also known that viruses like parvovirus 19, it's yet another virus that many people harbor, can be direct drivers of myocarditis. In our long COVID paper, there's literally a section called myocarditis in long COVID. I think that one of the things that we want to do is try to connect this trend in which these viruses that can drive myocarditis are reactivating and potentially see if by controlling their activity, perhaps with, herpes virus antivirals or other things, we could then limit at least some of the myocarditis cases. I'm not even saying that accounts for all of them, but it would be very strange if myocarditis wasn't driven by some of the viruses that are so connected to often causing it.
Thank you. Absolutely. That's really, it's really interesting to see how all these pieces fit together. Moving into the diagnostic space, because we have a lot of questions about diagnosis. So two-part question. The first is about getting the initial diagnosis. You described so many different mechanisms that could potentially lead to these long COVID symptoms that we're seeing, in addition to reactivation of these other reservoirs and other presence of other pathogens and antigens. So I just would wonder, in the diagnostic space, as a patient who thinks this might be their situation, approaches their doctor, what are sort of the key areas to zero in on when speaking to your doctor about your experience, and how can that be related to some of the treatments that you mentioned earlier? And then secondly, this is a question that's also come up from several sources, so thank you all again for your questions, about once you are sick, given that the CDC has changed the quarantine guidelines, what do you think about going back to work, and how should people go back to work after a potential diagnostic, and what did that journey potentially look like?
I don't think that was an intelligent decision to cut the quarantine time. I just honestly don't think that if people go back to work still sick, I'm not even sure that's going to help the workforce. It just seems like more people would get ill. I don't understand it as a policy. I do think that if you do have to go back to work under those rules, that the most important thing you can do is to wear a high quality mask and N95. Honestly, variants evolve and change. And to a degree, the vaccines hopefully protect to a certain extent against severe illness and hospitalization. But N95 masks actually prevent viral particles from going through them. So most of the variants, if you have a well-fitted N95, irregardless of the variant, should not really be able to get through your N95 mask. That is just a clear and obvious thing to do if you're in your workspace. I would hope it would allow you to do that. That's something that I do. I usually wear 2 masks, an N95 and a mask over the mask. Sometimes it depends on the situation. Some people even wear goggles. I personally don't even care if people think I'm crazy. I can wear goggles. Let's say I was on a plane, because if there's high viral load, the SARS-CoV-2 virus can infect the eye. And I'm not saying everyone has to do that, but if it comes down to it and you think you're at risk, it's something to consider. So those are some of the issues of just the workplace. And then getting back to being diagnosed with long COVID, which I think was your other question. And how do you approach a doctor about that? I don't have a perfect answer for patients because I'm not an MD either. So, I talk to a lot of MDs that are in this space. I do think that there are MDs who are already a little bit more aware of infection-associated chronic disease. Those would be people who have already been seeing patients with ME/CFS, even patients with post-treatment or chronic Lyme disease. I might approach a physician who's already dealing with those kinds of patients because they are open, at least understand the phenomenon of patients getting sick after an infection. I do think that the extent, it depends on the doctor, the extent to which you can bring them paper in the literature showing concrete evidence. Some of the papers that I even just showed in this talk, showing evidence of certain phenomenon, for example, that patients, have less oxygen extraction from tissue, which is a Harvard study. Things like that can help provide your doctor with some clarity on what to do. That being said, one of the challenges we face, I think we're going to have to innovate diagnostics. It's high time that we do, because the majority of conventional blood tests that are done in a doctor's office do not pick up on most of the trends I just talked about. You cannot just pull drug out of a little bit of blood out of your left arm and hope that it just shows all of what's occurring in the body. One of the biggest problems being the possibility that there's SARS-CoV-2 reservoir. Like I explained, the virus wouldn't be expected to persist in your blood. It would be in your tissue. So that's why it's so important that we begin to collect tissue and biopsy samples from these patients, because we can't look. It's silly to look for the virus in a place where it wouldn't even be expected to be, but that happens more than you'd think, where someone does a nasal swab test and they say, oh, you're negative by nasal swab, you must not have the virus at all. So to the extent that, those narratives can become more clear and you can, talk to your doctor about that possibility, it's open. But again, that's where we have to do the research to better show clinicians that there are clear examples of that these trends may be occurring, because at the end of the day, clinicians need clear research papers to draw from for the guidelines that they use.
Absolutely. Wow, that is so key. And just as a for those of you who are looking for resources and are struggling with your personal health journey, at solvelongcovid.org, there's a link to a informational center that's for patients with ME/CFS, patients with long COVID, and resources that are available just to help you guys in commonly answered questions. So again, for those of you looking for your individual health journey, solvelongcovid.org might be a great place to start your research. A couple more questions as we still have a few moments left. One question that I thought was really interesting and is a theme that's come up in a lot of different questions is about Omicron and Delta and what their relationship is to long COVID. Are these variants more dangerous for long COVID? Are they less dangerous for long COVID? What is your opinion about the influence of specifically the highly spreadable variants and their impact on long COVID?
Yeah, that's a great question. And I don't have a clear answer. Anyone that says that they know that Delta or Omicron could cause more long COVID, it's impossible to know at this point. We just need more data. That being said, one of the key things to understand is that, long COVID is not just being diagnosed in patients who have experienced severe disease or were hospitalized. A seriously large number of patients who had mild or even asymptomatic cases are developing long COVID. In that case, it's unfortunate that even if Omicron drives more mild disease, then that doesn't necessarily mean that there might be less long COVID, right? And so, it really depends, if it depends on what might be happening in a given patient with a long COVID diagnosis. If it's a patient, I mentioned that organ injury may just be, some patients may have been hospitalized and just experienced a brutal case of acute COVID, and they may still have chronic symptoms from that. They may get a long COVID diagnosis. And in that case, we may have less of those patients with Omicron because it may not be driving such severe inflammation during acute COVID, although, even that still has to be fully documented. But for example, because Omicron evades the immune response more, I sometimes worry that it might be harder to fully clear, right? Because the immune response needs to be robust to clear a virus from tissue. So perhaps there might be more problems with the persistence of SARS-CoV-2 with Omicron, right? So there may be different variables with the different variants that may impact long COVID, but overall, I definitely think that we should not let our guard down. I don't think anyone should be saying that long COVID isn't possible right now based off any variant. No matter what variants are out there, I think people should be very careful and very aware that this might occur.
Absolutely. As we continue to follow this evolving field and more data comes to us, I think a lot of those answers will become clear. But I think you're right to say at this time, we don't really have enough information. And that's why research is just so critical. Last question. This one's getting into the gut now. So we had quite a few questions that were very interested in the gut reservoir of the viruses that we were discussing. So this one person asks, what could be the role of stool testing in terms of tracking the progression of long COVID and COVID in general? They point to a study that was done early in the pandemic when sewer testing was being done to determine viral load to see where the virus was concentrated geographically across the country. So that's the first question. And the second question is, can stool testing be used for individual patients as a way to determine if the virus is still present, even though blood and nasal swabs might still come back negative?
That's an excellent question. And one of the things I worry about is that when the two examples I gave of intestinal tissue, one in which SARS-CoV-2 protein and antigen, or RNA and antigen, were in intestinal tissue, and one in which enterovirus and ME/CFS had been found with protein and RNA in intestinal tissue. In both cases, it was the tissue of the gut lining and not within the mix of organisms in the gut itself. So a stool sample may not necessarily even represent those organisms in tissue. Now, I'm not sure of that, but it's something that you have to consider. So one of the things that teams are doing, and again, I mentioned that we're trying to help a team get a study off the ground where they would collect the tissue via colonoscopy from patients with long COVID. They can also test stool sample at the same time, right? So they're going to actually do that. And they're actually going to be able to try to answer that question, which is, if they find the virus in the intestinal tissue, are they able to find it by the more routine stool testing as well, right? So it's an open question, but again, I cannot, actually guarantee that stool testing would help. Certainly it might be better than just a nasal swab test, but I'm not sure.
Interesting. So who knows, maybe at home stool testing might be the next generation of COVID testing. Fascinating. Well, that wraps up all the time we have for questions today. Amy, thank you so much for your time and sharing your wealth of knowledge on this topic. Clearly, we are just beginning to scratch the surface of long COVID and its impact on our bodies and our scientific fields in general. We can expect more ripple effects in the future. For those who are interested in asking questions about long COVID treatments, I highly recommend our upcoming webinar on January 27th about Ampligen, one of the drugs that's currently being considered for long COVID and ME/CFS treatment. For more information about that and the other resources I mentioned, please visit us at solvelongcovid.org. Bill, thank you again for being on the forefront of these critical issues and helping us bring them to light. It's been truly a pleasure.
Well, Emily Taylor, that was just a masterclass in moderation, and in the words of our moderator, wow. Amy, just I really appreciate your spending the time with us and the groundbreaking work that you and PolyBio Foundation are doing. Very, very important, whether it's on long COVID or ME/CFS, just tremendous. So thank you for being with us today. A fascinating conversation. And Amy, I hope you'll come back later in the year in this program, give us an update on the research and what you're finding. We'd love to see you again. Emily and Ovid, the GIC looks forward to this continued collaboration and partnership with you, with Solve ME. This is a very, very globally interdependent topic, and we're very proud to be associated with you through this process. So we look forward to continuing. So several people asked, let me just state before we close, the slides will be available on the GIC website along with the replay. So those should be posted tomorrow. And I want to just, before we close, I do want to say that we too are having a program on January 27th. So as soon as you're done with the Emily and Ovid program, come on over to the GIC program on January 27th. It's going to be a discussion on climate change and monetary policy. And we'll be joined by two fellows of GIC College of Central Bankers. That's Christian Noyer, former governor of the Banque de France, and Katarzyna Jadot-Kowalska, who's a former member of the management board of the National Bank of Poland. Again, that's January 27th. That's at 12 noon Eastern. And this event will be hosted by Peter Gold, the chair of the College of Central Bankers. So to register for that event, to see a replay of today or get access to the slides, you can go to the GIC website at www.interdependence.org. And as we conclude, we encourage you to take just 30 seconds, respond to our audience survey. Your feedback's very important to us. We appreciate you letting us know how we can improve the programming. We thank you very much for your attention. And until next time, good afternoon.